So this is where we kind of left off from last time. And from last time, we had just gotten done talking about some of the words, the vocabulary that goes with the cell cycle. And this time we're going to look at the cell cycle itself. And something important to remember is that the cell cycle or the life cycle of a cell is a lot more than just mitosis. So if we have this picture kind of we're going to use to refer to as the cell cycle, we're going to pick up with a cell right here, right after it's been made as a new daughter cell from mitosis. And at that point, it really has kind of two pathways it can take. The first pathway is if we continue kind of going around this cycle. So after a new cell is made, it will continue to grow and mature and then divide again right away. So in that case, it goes through a number of we call phases, like the G1 phase. It's called G1 for gap 1. This is a period where the cell is growing and doing any work that it needs to do depending on the type of cell it is, and storing up resources for what's called this S phase, and this is where, or synthesis phase, and this is where the cell makes a copy of or duplicates its DNA. And then after that phase comes the G2 or the gap 2 phase, where it's continuing to grow and store up resources to prepare for mitosis, where it's going to split again. The other option, all right, this first option is kind of what cells that divide consistently go through. And the second option is it might grow and then take this pathway off the cell cycle to the G0 phase, or like the resting phase, where it is kind of in a state of just not dividing, right? And some cells, like brain or nerve cells, never divide again. You know, they never go through mitosis and produce new ones, right? Some others will kind of stay in this phase until something prompts them to get kicked out, like a liver cell. So if there's damage, the liver cells can regenerate, your liver can regrow, right? So it might be, a liver cell might be in this G0 phase, and damage might kick it out back into the bigger part of the cell cycle where it's going to start to grow and divide again. If we look at this whole thing, we can kind of group this cell cycle into two pieces. Interphase, which is everything else besides mitosis. All the other stuff here we can group into interphase. That's when cells are growing and replicating their DNA and making new organelles and new biomolecules. And then mitosis, or the mitotic phase, is the actual process of the cell separating, dividing its chromosomes between two new cells, dividing the organelles in the cytoplasm between two new cells. So everything except the M part we can lump together and call interphase. All right, interphase is not a phase of mitosis. It lies outside of mitosis. It is a phase of the cell cycle, though. All right. We're actually going to talk about control of the cell cycle in some separate videos on Monday, so I won't go through this part now. I should have taken this out, but there are certain things that control the cell cycle and whether the cell passes into one phase or stays back in the other, but we're going to talk about that separately on Monday. So like I said, in this picture kind of shows another representation. Interphase is everything outside of the mitotic phases. The cell spends about 90% of its life cycle there doing everyday jobs, all right? And in cells that are in interphase, they kind of just look like normal cells. They have a big, well-defined nucleus, and the DNA in the nucleus kind of looks like long, stringy material, because it's just chromatin fibers packed together. And there's not really anything remarkable about it. So if you'd look through a microscope at a cell, most of them that you would see kind of motionless in on that slide would be an interphase. We had already kind of talked about the three phases called G1, gap, first gap, S, the DNA synthesis, and G2, second gap phase in interphase, but we can go a little bit more in depth with them right here. So the G1 phase, the first stage after cell division, the cell's kind of recovering from division. It's growing rapidly, and it's reproducing its organelles, so it has enough to do all the cellular work. And it's doing kind of everyday cell jobs. At that point, cells can break off into G0 phase, ones that don't need to complete the cell cycle, um, or sorry, ones that might leave and just be resting temporarily. They can break off into the G0 phase, where they're continuing normal cell operations, but they're not at all preparing to divide again. Um, and a lot of cells that enter the G0 phase will just stay there until the organism dies. But others, like we said, like the liver cell, can be stimulated through some event to re-enter the cell cycle. The S phase, like we talked about, is where DNA is being copied. The chromosomes duplicate, preparing for mitosis, because you can't go through mitosis unless you have twice as much DNA, enough for both new cells. And then right before mitosis, there's a second stage of growth and preparation um, and acquisition of resources for mitosis. So these pictures are from your book, using them because they're most consistent to what you'd see in your book. This kind of is what the cell would look like in the G2 of interphase, or just interphase in general. Got a nice nucleus in the chromatin fibers, and then our centriole has duplicated. So remember, we have like those two centrioles. These are going to be what's going to help us in the mitosis phases, or mitotic phases. And then there is the small 10% of the cell's life cycle that is the mitotic phases, or mitosis. And this is when the DNA needs to divide between the two daughter cells. And mitosis is made up of these five phases: prophase, prometaphase, metaphase, anaphase, and telophase, in that order. And so we're just going to briefly go over each. I'm just going to mention kind of the important things about each to kind of keep in mind. So here's just a little bit of an overview. So in prophase, and in each phase we talk about, you can kind of see what we're talking about because there'll be a little picture from your book right here. The chromatin, or the DNA, condenses so that then from a microscope there are visible chromosomes with chromatids. So now you can see visible chromosomes. It's a lot dense, well defined. These centrioles, centrosomes, small tubules called spindle fibers start to extend from them. And that starts in prophase. And they start to move to the opposite side of the cell, all right? Because each opposite side is going to kind of turn into one of the new daughter cells. The nuclear membrane starts to break apart, so the nucleus disappears. And the newly condensed chromosomes just start to exist outside in the cytoplasm. So that is prophase, preparing for division. In prometaphase, these long spindle fibers, tubules that is coming from the centrosomes, start to attach to proteins on the centromeres, the central part of the chromosome. And those proteins that it attaches to on the centromeres are called kinetochores. So that's the protein it's attaching to on the centromere. Now, since they're attached to the centriole, the chromosomes start to move around and just begin to arrange themselves. You can kind of see it here, and you can see it definitely in the high contrast microscope image up there. So here is the kinetochore protein on the centromere of the chromosome, and the spindle fibers from the centriole attaching to it on each side. So, like we talked about, there's one centrosome over here, and its spindle fibers are going to attach to all of the chromosomes on this side, and the other centrosome over here, its spindle fibers will attach to all of the chromosomes on this side. In metaphase, those spindle fibers that have attached to the kinetochores line up the chromosome along the middle of the cell, called the metaphase plate. So all of the chromosomes are now lined up along the metaphase plate. And like we were talking about before, there's one spindle fiber from each of the two centrosomes that attach to opposite sides of the chromosome. So this centrosome has spindle fibers going out to the closest side of each chromosome, and this centrosome has spindle fibers going to this side of each chromosome, just like that. In sorry, I got ahead of myself. I apologize. So just another picture, microscope images of metaphase. And then in anaphase, those spindle fibers start to shorten and basically rip the chromosomes apart at the centromere. So the sister chromatids, each half of each chromosome, duplicated chromosome, moves to opposite side of the cell. They're being pulled by motor proteins attached to these tubules. And they're getting pulled to opposite side of the cell. As well as that, this cell starts to elongate or turn ovular into an oval. So the poles start to push farther away from each other. So in anaphase, away, the chromosomes move away from each other. And so here's a little picture of the anaphase at the centromere, the two sister chromatids being separated from each other. Separated from each other. You can see another picture kind of anaphase. And the kinetochore, the motor proteins that are moving the sister chromatid or this chromosome down towards the centromere. And then in telophase, the chromosomes have arrived at opposite sides of the cell. Look at how they're at opposite sides of the cell in both these pictures. At this point, two new nuclei start to form around each of the sets of DNA that are now on the opposite sides of the cell. And the chromosomes start to disperse, so they become back into long, stringy chromatin, so that they're no longer easily visible under the light microscopes. As you can notice in this picture, those spindle fibers start to go away, and the centrioles no longer have those long projections. And then the cytokinesis process is going to start, where actually the cytoplasm and the organelles begin to divide. So lastly, we have cytokinesis. So in animal cells, this pinching off of the membrane right here by this arrow, it's called a cleavage furrow, starts to form, right? And it's pinching the membranes apart, kind of into two pieces to form two new cells. So kind of like if you are tightening a drawstring, it's kind of the same thing that's happening here, except it will eventually pinch them completely in two. And so here in an actual uh, microscope image, here is a cleavage furrow being created. So it forms here, and then eventually two new daughter cells. And that's going to be the end of mitosis. That's going to be, now we have two new daughter cells, and they can go into G1 right after this and go about their way on the cell cycle.